Welcome to Learn Biology, and last time we were discussing cell parts and we left off on the endoplasmic reticulum. So we said that the endoplasmic reticulum is split into two parts, smooth and rough. So the smooth is used to make fats or lipids, and the rough endoplasmic reticulum is used to make proteins using ribosomes. And what we need to understand is that there are few other parts that are related to the rough and smooth ER. But first we'll discuss Golgi bodies or Golgi apparatus. So the Golgi apparatus is 8 membrane-bound channels which appear to be flattened sacs. So we'll change the color here to something such as yellow, and this is what the Golgi apparatus looks. So it's 8 sacs. And 8. So what the Golgi apparatus does, and let's just change the color here, the Golgi apparatus does is it transports and refines molecules. So transports and refines molecules. Transports/refines. And how this works is that, let's say the smooth ER has just made a lipid. So we'll take this tiny blue thing, call it a lipid. So now we have a lipid right there, and the only way that lipid can leave the cell is by the Golgi apparatus. So what happens is that if the ER, rough ER or smooth ER, is close to the Golgi apparatus, in this case, if the smooth ER is close to a Golgi apparatus, a tube forms between the Golgi and the smooth ER. And this tube is called a cisterna. So cisterna. And what it does is transport molecules to the Golgi apparatus. So now this Golgi apparatus goes in and gets refined and refines this lipid. So it goes in and is refined. Then what happens is that the Golgi literally takes a piece of itself and it branches off. So let's make sense here. So let's say right here is where the lipid is. Now what happens is that the Golgi branches off. This section will branch off and will go. So now this section will branch off with the lipid and leave the cell. And then it comes back and hooks back together. However, let's say here's a rough ER. So this is a rough ER and the Golgi apparatus is far away. What happens then is that a transport vesicle, this little tube, carries a protein that's made by the rough ER and takes it and then connects to the Golgi. And then this connection does the same thing. It goes through the Golgi, gets refined, and then the Golgi branches off and transports it outside the cell. So that is how the ER and Golgi apparatus work. Then we have lysosomes. Lysosomes are, we'll make them color here. So lysosomes. So lysosome. And what the lysosome is known as is a suicide sac. Suicide sac. What it does is digest substances and waste. If a foreign invader comes from outside, let's say a bad bacteria infects the cell, here we'll have a pink circular bad bacteria. So that's a bad bacteria, and it goes inside. The lysosome will literally go and eat it, and then it will digest it. So that is how the lysosome works. It is, and why it's called a suicide sac is, let's say the cell is getting old. the lysosome can it can digest itself and get rid of itself. So lysosomes are used to digest waste. So that's what a lysosome is. Then we have another cell part called a mitochondria. So we have a mitochondria, and what a mitochondria looks like is a ovular shape and has tiny little grooves within it. So that is a mitochondria. So the mitochondria, let me just change that here. The mitochondria is used to convert what the cell has into energy. So it converts food into usable energy for the cell, and the energy for the cell is known as ATP. So mitochondria is the site of cellular respiration. Cellular respiration is what converts food into usable energy. So ATP is made through cellular respiration. So cell respiration. So thank you for watching Learn Biology, and we'll be covering remainder of the cell parts in the next video. See you next time.